cases. And so if we think about different kinds of steps that one would take here, if you're building a metabolic pathway to a natural product, there's a question of if you know that that product just exists in nature, or if you know exactly how it's made. So if the natural pathway is elucidated, and that's what you want to use, then you can try to figure out, okay, in that organism, that pathway is coming from these central metabolites, and my organism, maybe an E. coli or a yeast, has those metabolites up to a certain point. And at this point, it no longer performs that kind of metabolic reactions. So then you're going to transfer everything that's downstream of that, the heterologous genes, into your organism. And then this is still really oversimplified because there might be a variety of challenges that you have in trying to express those proteins correctly. There might not be the right cofactors available for those steps. Those might be localized to the wrong part of the cell. And so there's potentially a ton of troubleshooting that may need to be done here. And that generally has to do with how, from a phylogenetic standpoint, how distant is the organism that has that pathway from the organism that you want to have the engineered pathway. If your natural pathway is not elucidated, then you have potentially many additional steps. And so this is an area that generally falls outside of the metabolic engineering core community, but has increasingly become more important in order to identify and make products that are new and innovative or needed. So one way to do this, and we'll spend more time talking about this in the next lecture, is to focus on genes that seem like they're likely to form a pathway towards your product. That can be based on sequence similarity and other techniques, including clustering of genes within biosynthetic gene clusters. So you can go the route where you look at these genes and try to figure out then what are the function of the proteins that they encode and try to piece together a new pathway. Or if, depending on what your organism is, what your pathway that you're looking at is, you could try to do more metabolomic analysis, focus on the actual metabolites and small molecules. And often you'll want to do these approaches together. But if you go with a metabolite-oriented strategy, if you can detect small concentrations of precursors, which you aren't going to know offhand are precursors unless you have a good idea of what the reaction rules might be for your pathway. But if you imagine you have this product and maybe it has a particular kind of functional group, and based on nature, you know that generally there's this kind of functional group prior to that in a lot of pathways, then you can try to, based on the biochemical intermediates that you identify, if you see that, you can then try to identify what kind of enzymes might be required in order to have that kind of a pathway. And there are other techniques here. Depending on what your organism is, you might be able to challenge it. You might be able to essentially spike in a molecule of some kind that's related to your pathway, and then almost like if it were a tracer, observe the flux and what it gets transformed into. You can also look at the transcripts and see if certain pathway genes get overexpressed based on, if you see a gene expression shoot up, maybe that's part of your pathway, and then you look at that more carefully. And then you can also use techniques like knockouts. If you can modify your original organism, then you could knock out a gene, and then if you see that product no longer being made, you know that that's probably part of the pathway. So that is still under the framework that there is this product that you're trying to make that another organism does make. and that you want to just copy that. If you don't want to copy that, or if you want to build a pathway to a non-natural product, then you have to use a different approach. And this becomes reliant on this concept of retrobiosynthesis, which we're going to discuss more for the remainder of this lecture. And really, that's the difference, is that you do retrobiosynthesis, and then once you've kind of identified some candidate pathways, you just transform them, you transform your host with them, and then you do some troubleshooting on this pathway. And of course, in this case, both the troubleshooting and the retrobiosynthesis steps might not be trivial. So the way to also think about the relationship between all these different kinds of metabolic engineering is this sort of hierarchical framework here, different levels that were described in this review, of going from natural metabolism to a synthetic metabolism. And so, you know, the first kind of layer in here, so that both this arrow and this graph correspond to one another. They're from the same paper. And I think what's important here, and another way in which metabolic engineering and protein engineering really intersect, is this idea that you have a relationship between your pathway solution space and your enzyme solution space. So if you're operating in this sort of close to the origin and small solution space regime, you've got your natural pathways. And then what you can do is you can just copy and paste existing enzymes. And what you're doing then is you're not really expanding in the direction of surveying or creating new and interesting enzymes, but you are potentially creating new and interesting pathways just by taking the enzymes that nature has already given you and that has purposed for those kinds of pathways. You can go beyond that and then start doing some more mixing and matching to create really novel pathways. You know, here the difference might be that this was a pathway that didn't exist in your organism. So now it's in a new host, but now this is a new pathway, but it's still known reactions and known enzymes. And what can change then with engineering, especially at the protein level, is this idea of then getting synthetic pathways that have novel reactions, where you're still relying on known enzyme mechanisms in this case, and then taking that further and going to entirely new reactions. And so you can see how these bubbles here are expanding the solution space, both in terms of the enzyme direction, but also in terms of pathways. Because essentially the diversity of pathways that you can assemble to different metabolic products is entirely related to the kind of enzymes you have available. And if you have the tools to generate new enzymes to make your desired reaction something that doesn't exist in nature, then that just makes the field of metabolic engineering even more powerful. You can start to do more interesting retrobiosynthesis. So to dive into what retrobiosynthesis is in a little bit more detail, I want to just spend a small amount of time thinking about the different examples that relate to the plots that we just saw. So if we're in this column here from the same review, there are examples related to CO2 fixation. And you can see how if an organism, say, already has Calvin cycle and is capable of fixing CO2, then you can overexpress a transketolase. Then you could, even though this is not at all trivial, move, as a next step of innovation, you could move the Calvin cycle to E. coli. And that's actually pretty sophisticated, but that's still level 2. Going into the third level is taking CO2 fixation and taking known enzymes and putting them together in a different way. And that would be where we would start to describe this as de novo pathway design, because that's not the pathway that it would have been used naturally. So then, using more enzyme engineering techniques, you could potentially do this in a very different